is work that I did for my master's at Bucknell with Dr. Chris Martine. Right now I'm at the University of Pittsburgh. I'm starting my PhD. And this work was done with Angela McDonald, who you'll hear from later, and Scott Schutte with the Western Pennsylvania Conservancy. And I'm going to talk to you today about Baptisia australis variety australis, or its common name, blue false indigo, and how we use natural history and population genomics to assess the state threatened taxon. Oops, sorry. Okay. So I'm just going to walk you through an outline of what I'm going to talk about today. So I'll give you some background information on the species. I'll talk about granivory and some populations along the Allegheny River. And then I'll talk to you about population genomics. And I'll also talk about the conservation implications for all of this. And then at the end, you can ask me any questions. I tried to leave 10 minutes. So. Okay. So some background on Baptisia australis, variety australis. You might not have heard the name with the variety specified before. There are several varieties, but variety australis is the only one that occurs in Pennsylvania. So we're there at the northeastern edge of the range, you can see. And the species is in the Fabaceae family. It reproduces via rhizomes underground, but also via hermaphroditic flowers that are bee pollinated. And so if you have it in your garden, you might know it has these really big roots that are well rooted in the soil, and then they can send rhizomes out to create more of themselves. And they flower in late May and early June. They're really pretty flowers, as you might know, and they're perennials that come back every year, and they're popular in cultivation. Pennsylvania has a rather limited distribution naturally, so it's only found along 4 waterways currently: the Allegheny River and 2 of its tributaries, the Clarion River and the Red Bank Creek, and then the Youghiogheny River further south. And in part because of this limited distribution, the Pennsylvania Department of Conservation and Natural Resources considers it to be threatened, and the Pennsylvania Natural Heritage Program considers it to be imperiled within the state and then vulnerable globally. Other factors in this rank, in addition to that limited distribution, are the specialized habitat that it occurs in. So it occurs along those waterways, as I mentioned, and it occurs in these open prairie-like systems along riverbanks. And that open system is kind of facilitated by either big chunks of ice that come down in spring thaws, scouring the banks, or flooding. So it keeps trees from growing and shading them out. But this habitat's being lost for several reasons, including development by humans and damming upstream. Climate change is also a factor. So with warming climates, we'll see less of that ice scouring that is necessary. And then climate change also changes precipitation regimes, as I'm sure many of us know. They actually spent a good chunk of their flowering time underwater for one of my field seasons. So we got a ton of rain in May and June, and we actually couldn't access them until July, which resulted in very few flowers and fruits. So the plants were alive, they're still there, vegetative, but no fruits. And then invasive species, so here are some photos of phalaris or reed canary grass, and then knotweed, which co-occurs with Baptisia and a lot of the populations in Pennsylvania. This is a map that kind of gives you an idea of the historic distribution in Pennsylvania and in nearby states, and kind of gives you an idea of how much the distribution has declined. So I made this map based on herbarium records, and the darker green dots are more recent collections. So you can see there's only 5 counties in Pennsylvania that have fairly recent collections, and those are the 5 counties that we find Baptisia in currently in Pennsylvania. And so for my work, I wanted a population from outside of the state. For this genetic work, it was useful for me to have a population from outside of these samples. So I went to Ohio and looked for this population with Angela actually, and it's actually no longer there. So we have no more extant populations in Ohio. So we ended up sampling from individuals that were from West Virginia. We didn't actually go to West Virginia. Some of you might be familiar with Ernst Seeds in Meadville. They have a lot of native seeds. And they actually collected from a population in West Virginia and have been growing them in Meadville to produce seeds. And so Calvin Ernst was kind enough to let me come sample the tissue from those plants. All right, so that's the background I have for all of you. And then I have 2 chapters to my thesis, and I'm gonna highlight some different aspects of it. So this first chapter is about granivory and a subset of my populations. And so this focuses on the larval form or the caterpillar form of a moth that is known to eat the seeds of Baptisia and other parts of its range, but hasn't really been documented in Pennsylvania. So my first question was, is this Lepidopteran larva, is this caterpillar present in Pennsylvania? And if so, is the number of seeds negatively impacted by its presence? And so I subsampled these 5 populations along the Allegheny River. And then my second question was, could we correlate the damage of the plant or the fruit with factors that could help us predict what sites or plants might be susceptible to these caterpillars that are eating our seeds? So in the field, we collected 15 fruits from 5 sites along the Allegheny River. We recorded any damage of fruits. And what we think is happening here is that these eggs are being ovideposited into these fruits, and when they hatch, the young then eat the seeds, and then they eat their way out of the fruit once they're big enough. So we recorded any kind of damage like that, the height of the plant, the number of racemes or shoots that come up for each individual, and number of fruits, distance to the river, those sorts of things. Back in the lab, we dissected the fruits and recorded the number of seeds within them, both healthy and aborted. So we noticed this interesting trend where if a granivore was found in a fruit and it had started eating seeds, sometimes the seeds would become kind of shriveled up and essentially be no longer viable. So they wouldn't look like they were healthy seeds anymore. The plant would kind of abandon putting resources into them. So we counted the number of both healthy and aborted. We recorded any evidence of insects, such as the insect itself, or partially consumed seeds or frass, and then the length of the seeds. And we utilized some generalized linear models to look at the statistics. And instead of showing you like big tables of my results and p-values, I'll kind of show you this figure that kind of sums up what we saw. So essentially, is the number of seeds negatively impacted by herbivore or granivore presence? The answer is yes. So you can see if frass is present, we often have fewer seeds. So the key conclusion here is the number of seeds is significantly negatively impacted by damage or presence of frass, which is kind of what we would expect, but no one had really shown that for Pennsylvania before. So it's important information to establish. And then our second question was, is the damage of the fruit or plant correlated with anything that could help us predict what plants or sites might have these granivores present? And the answer again is yes. So this is again kind of intuitive, but maybe useful for practitioners out in the field. And so damage is actually correlated with the presence of physical damage. So you looking at the fruit and saying, oh, there's a hole there, looking at the leaves and saying, oh, they've been partially consumed. So those sorts of things. And that could be helpful for practitioners going out in the field that don't necessarily have time to collect fruits and take them back and open them up and look for granivores. They can simply do a quick visual survey and say, about half of the plants look like they have some kind of holes in the fruits. So that means the granivores are present here and likely impacting the number of seeds. So our key conclusions for this chapter were that granivory is present in these Pennsylvania populations of Baptisia australis, variety australis, and that these visual assessments could be useful in assessing the degree to which populations are impacted by these granivores. In some cases, granivory can significantly reduce the seed count, and we recommend including granivory as a factor in population assessments and conservation calculations. So this is my second chapter, and this is my population genomics and the implications for conservation. And I had lots of questions that went along with this chapter that I could talk about, but I pulled out the one that I think is maybe most interesting or most applicable and we got the kind of the coolest results for. So this question was, how many genetic groups are in Pennsylvania? And we suspected maybe 2 genetically distinct groups. This one up here with the Allegheny, the Clarion, and the Red Bank Creek, and then the Youghiogheny down here. And this is kind of important for management in the sense that whenever you have a species of concern, it's often split up into these management units or element occurrences depending on what organization you're talking to. And those groups are important for managing the populations. And often these are kind of split up just based on human constructs like waterways or physical distance, but that might not necessarily accurately reflect the actual populations that are genetically present, so indicative of breeding between individuals and groups. So it's important to answer this to help establish these management units appropriately so we can help to manage things the way they naturally occur. So I'll walk you through my methods. I'll try to keep it brief and fairly interesting, and then I'll talk about the data analysis and show you some results, and then conservation implications. So tissue was collected from 24 sites, which resulted in 326 samples being sent out for DNA sequencing. And this was most all of the sites known in Pennsylvania where Baptisia australis occurs. And to get these samples, I paddled about 70 miles and drove nearly 5,000. So very thorough sampling. And then at each site, we collected tissue from 15 plants per site. And I guess whenever I'm saying that I collected tissue, it's as simple as plucking one of those leaves off. So those 3 little leaflets, I plucked one of those off and put it in an envelope to dry down, and that's what we use to get DNA out of. And then we sampled from 1 meter apart or greater when possible to try to avoid getting that same individual, because again, they reproduce via rhizome, so we don't want to just collect the same individual that was connected underground. And here I should acknowledge that our partnership with the Western Pennsylvania Conservancy and the Natural Heritage Program through Dr. Ph.D, who's an author on this talk, was really critical in getting this work done. So because it's a species of concern, you can't readily access the locations that it's found, and you can't just go out and collect it. You have to have special permits. So working with Scott allowed us to do this work. So back in the lab, we extracted with a kit, we ground up that tissue and extracted DNA, basically following a recipe. We did something called reduced representation genome sequencing, which is a mouthful, but essentially it means we cut up the DNA with enzymes. So we had this DNA we extracted and we cut it up by adding essentially an enzyme to the mix, which results in lots of tiny comparable pieces of DNA. So essentially this enzyme is cutting at the same place in all of our samples and giving us all these tiny pieces from about the same place in all of our individuals. So we can compare those pieces across all of our samples. And what we ended up with was about 800 million of those tiny little pieces, or SNPs as they're called. And then we processed those. So not all of those are great quality and not all of them are common to all of our individuals. And so we had to pull the pieces and filter them. And we ended up with about 350 pieces that were comparable across most of our samples. And then those pieces were used for a lot of our analyses. And we had 318 individuals in our final data set. So we only kicked out about 7 individuals, which is pretty good. And then we analyzed the data, all those little pieces, to calculate the number of populations and lots of other things. But again, I'll just be talking about the number of populations, genetic populations. So this first figure is a PCA, or principal components analysis. And these axes, or PCs, don't really mean anything super concrete. It's just a way to show variation amongst individuals. So each of these little dots is one of our individuals that we sampled, and they're color coded by the sites that we sampled. And so here we have the Allegheny River. All of the individuals are blue from the Allegheny, and I tried to color code them from upstream to downstream, with the upstream populations being a lighter color and the downstream populations being darker. And this is just essentially showing differences in separation between the individuals based on that genetic data. And so here's all those Allegheny individuals. And then next would be the Clarion individuals, and they cluster together as well, further away from these Allegheny individuals. And again, to remind you, the Clarion River is right here feeding into the Allegheny. And then we have the Red Bank Creek populations. So there were 3 sites that we sampled, and they're color coded here. And you can see they cluster together, but they're also clustering pretty closely with some of these downstream Allegheny River populations, which kind of matches up with the spatial distribution that you see in the map. And then the Youghiogheny population way down here by itself, clusters by itself over here. And then those West Virginia populations that were from Ernst, they're clustering together by themselves, so that makes sense. This next figure I'm going to show you is pretty similar. It's a discriminant analysis of principal components. Again, all of these dots represent one individual, but here the interesting part is this analysis tries to find the number of groups indicated in the genetics that we're giving it, the data that we're giving it. So instead of just taking the populations that I sampled or the sites that I sampled and using those to color code, it finds the number of populations it thinks is present in the data, and then it plots them based on differences. So again, I have this map to kind of show you where things are, and a lot of these populations cluster together. We have the Youghiogheny, again, clustering by itself, and West Virginia up there by itself, which makes sense. They're pretty far away from all the other populations that we have up here. And although all of these are clustering really closely together in this figure, the program still thinks that they're all separate groups. So we have the Clarion River, again, right there, and then the Red Bank Creek, it found as its own kind of genetic population, so down here. And then it split the Allegheny River into 3 different groups. So we have the upper Allegheny, so these populations are sites above the Clarion River, the lower Allegheny, so those are the sites that I sampled between the Clarion River and down here where the Red Bank Creek meets the Allegheny. And then we have this one site down here all by itself, downstream of the Red Bank Creek, called River's Edge, and the analysis found that it was its own kind of genetic thing. And here's another analysis that tells us something a little different. It does something similar. It's an SNMF plot, and it tries to find the number of K, or genetic groups, and it found something similar, but it found 2 less groups, so K of 5. And what all of these bars indicate is the likelihood that an individual belongs to a population. So each of these vertical lines, which are kind of hard to see because there's a lot of them, the likelihood it belongs to a population. So the more brown a bar is, the more likely it came from West Virginia, the more green, the more likely it came from the Clarion River. And so this all makes sense. The West Virginia individuals are from West Virginia, the Clarion individuals are from the Clarion River, and the Youghiogheny individuals are from the Youghiogheny, matching up with the last analysis pretty well. And then we have all of these individuals, which are a lot more confusing. And so what was more populations in the last analysis is now just broken down into upper and lower Allegheny. So the upper Allegheny is its own kind of thing, similar to before. It has a lot of that dark blue in it though. And then we have the lower Allegheny, which now includes that River's Edge population, which looked like it was its own thing. And it also now includes the Red Bank Creek as well. So it's kind of absorbed 2 populations. So largely mirroring the last analysis, but a little bit different. Not getting a clear picture yet. So I'll show you one more analysis and then we can talk about what we ended up deciding all of this means. So this is a heat map of FST, or gene flow. FST is a measure of gene flow, and I'm sure the other folks that are talking today will talk a little bit about it. But FST greater than 0.15 means significant differentiation. So that means populations that have FST greater than 0.15 are not sharing a lot of gene flow. They're becoming separate entities. And so this is a pairwise comparison, and I've tried to label all of the waterways. So here we have West Virginia. When you compare the West Virginia stuff to any other population that I sampled or any other site that I sampled, there's significant differentiation. And the same is true for the Youghiogheny River. When you compare it to any of the other populations, it's significantly differentiated. It's not sharing much gene flow based on this color indicator over here. And then with the other waterways, those values, like they're a little bit closer to the 0.1 or 0.2. So they're approaching that significant genetic differentiation value. They might not be quite there yet, or they might be there yet. The Clarion looks like it might be at around 0.2, but those are more around the range that we're not sure if they're differentiated yet, but they definitely could be. And again, the river's edge is a little bit darker, so you can see it's kind of its own thing along the Allegheny as well, kind of mirroring what we saw in that one scatter plot that I showed you. So that was a lot of stuff that I just talked about, and none of it really agreed. So I'll recap what all of it kind of meant. So that PCA I showed you, the first one, with all the different colors, indicates genetic differences between populations on different waterways with an upstream-downstream pattern on the Allegheny, which I think is pretty cool. And the DAPC shows 7 genetic groups, and then the SNMF shows 5, and the FST kind of shows patterns of genetic differentiation on waterways, but not always super clear. So to draw a conclusion from all of that, how many genetic groups are in Pennsylvania, we suspected 2. In the end, we decided that we could definitely say there were 3 groups, based on our data at least. So the Clarion River always clustered out by itself. In all of our analyses, it showed up as its own kind of genetic group. And so we decided to declare that was the case here on this map. And then the Youghiogheny River, again, being so far south, definitely its own thing, definitely always clustering out. But then, especially with that one bar graph I showed you, it kind of looks like the Allegheny River and the Red Bank Creek are kind of their own big mixed population. But we do acknowledge that there's this binary genetic structure occurring. So we can't really say these are all genetic groups because they are sharing some gene flow, but not necessarily as much as they maybe once did whenever populations were more continuous. And so we have these 4 kind of subpopulations: the upper Allegheny, the lower Allegheny, the Red Bank Creek, and then the River's Edge population. So what are the conservation implications for all of this work? Again, this was in partnership with the Natural Heritage Program and Pennsylvania's DCNR. So this was actually funded by a grant from DCNR. So we're working closely with both of those organizations to share the information that we've gathered. And then the Natural Heritage Program can utilize this data for their NatureServe rank calculator. So I mentioned that this is an S2 species. They can reevaluate those S ranks with that calculator. They also have a climate change vulnerability calculator. They can take into account some of this data and look at the vulnerability index for Baptisia in Pennsylvania. And then again, those management units and element occurrences. So understanding how many groups are present in Pennsylvania and how we should be managing them, not just based on man-made factors like distance or waterway, but more based on the actual genetics of the populations. So what are the takeaways that I want anyone that listens to this talk to have? It was a lot of things. What are the main things that I think are important? Granivory is impacting these Pennsylvania populations, which isn't something that was necessarily considered before. So it's important to maybe consider in the future, and whenever we talk about the conservation of the species, the granivory that it's experiencing is maybe a problem that we haven't really thought about before. And that there are 3 genetic groups in Pennsylvania, and they should be managed as such in the future to try to maximize the way we conserve the species. And maybe within these groups, that developing structure I talked about, especially along the Allegheny River, there's this developing structure, so maybe facilitated gene flow could be helpful. Because Baptisia reproduces via seed, it could be fairly easy to collect that seed and spread it, and kind of try to restore the gene flow that maybe once was whenever these populations were more continuous along the rivers before people started developing and damming. And with that, I have lots of folks to acknowledge, including our funding sources, the Pennsylvania Department of Conservation and Natural Resources, Bucknell, the Carnegie Museum of Natural History, the Botanical Society of America, and the Southern Appalachian Botanical Society. Again, the Western Pennsylvania Conservancy and the Pennsylvania Natural Heritage Program Partnership, my lab at Bucknell, and all of the other great folks at Bucknell as well. Now with that, I would be happy to take any questions.